The last type of particle transport is active transport. So this will be covering this section of your notes that's highlighted in yellow. So when we're talking about active transport, we're talking about going against the concentration gradient, going against the flow. So it's kind of like trying to swim, trying to pedal yourself upstream. So when you're going from this low concentration into a high concentration, it does require input on the part of the cell. So it is costly to the cell. It requires energy. And when you're talking about active transport, you're also going to talk about the need for the help of facilitator, which would be a protein. And in this case, we don't call them channels, we call them pumps. And if you think again about the word pump, a pump implies the use of energy. So as you look at this protein pump here, we are going to make it in green. We see that it looks kind of sort of like our channel protein, so it is going to provide a passageway. But because of the concentration gradient, it's going to require energy. So if you look in this case, we're going to be transporting glucose molecules. So you can see this kind of ring shape of a glucose molecule. And at the bottom here, we have a lot of glucose molecules. So there's a high concentration of glucose molecules already inside of the cell. But for some reason, our cell still requires more of them. So now we're going to take glucose molecules and force them into an area that is already highly concentrated. And this does require energy. So when you see this capital E here, this is a way to represent energy is with a capital E. Now, there are two examples of active transport. One is called endocytosis and the other is called exocytosis. So if you just look at the words tell you a whole lot about the action. So endo, inside, and cyto cell. So inside the cell. So we can see here that the cell is bringing in some materials inside of the cell. Well, there's a whole lot already going on inside of this cell. So we're bringing in substances into an already highly concentrated area, and that does require energy. Now, you also see endocytosis with the action of antibodies that will engulf foreign invaders, pathogens, viruses, bacteria, and then destroy them. Now, the opposite of this, we can see exocytosis. So, exiting, going out of the cell. And so we can see an expulsion here of materials. So, this could be proteins that are going to be used somewhere else in the body. And so we can see that these materials are being taken outside of the cell, the same way we kind of remove waste products. We could also remove waste products from our cells. And both of these are going against the flow from the low to the high concentration and will require an input of ATP energy. Now, if we look at all of these together, we have on the left side representation of our passive transport. So we have simple diffusion. These little guys here can just go straight across this membrane because they're small and because they're nonpolar. These guys are a little bit larger, they're polar, so they have to have some passageway to kind of allow them to go through all of these fatty acid tails without some type of reaction with them and just to move them to the other side. And then we see a completely opposite situation. So we're not going with the flow here, what seems natural, from taking a whole lot where there's fewer until we level it out, where we reach homeostasis or equilibrium. Now we're taking just a few of these and we're forcing them over into a side of the cell that is already full of these. It's already highly concentrated. And that does require the use of a protein, and these are going to be pumps, and also the use of ATP to operate these pumps, to operate the movement of these molecules against the concentration gradient.